Radhe Radhe, good evening. Haji, welcome to another video tutorial. Today we will discuss about the genetic material, how we can decide that the genetic material is DNA, which type of experiment is performed here, and finally how we conclude that the DNA is the genetic material. So be in touch till last of the video and let us discuss one by one. First of all, a experiment is performed for the search of genetic material, which is known as the transformation experiment. Transformation experiment. In this transformation experiment, a scientist named as Frederick Griffith in 1928 used a mice and a bacteria known as Streptococcus pneumoniae. He used this bacteria for his experiment, and he used actually this bacteria have many stains. He used two stains. One is S third type, who have a capsule and a shiny surface. And the second he used, that is a stain, is known as R2 type, who have not capsule and have rough surface. The S type or capsulate type of stain, this stain S type or capsule type stain have the ability to cause pneumonia. That's why it is also known as virulent. S type is a virulent stain, while the R type does not cause pneumonia, and that's why it is known as non-virulent. Non-virulent. So from this it is clear that the S third type stain, or simply we can say that the S type stain have virulent property and it may cause pneumonia to the bacteria, mice, and the R type stain don't have a virulent property. That's why it is known as non-virulence and does not cause pneumonia to the mice. Okay, now let us discuss one by one. So what he do? He use the R type of stain to check whether the virulency property is present or not. He use R cell or R type of bacteria and inject into mice. The live mice will be alive. He used S type stain and inject into mice. The mice will died due to the pneumonia. Due to pneumonia. Again, he used heat-killed S-type cell or bacteria and inject in the mice, and the mice will alive. Now, in further, he used again R type as a normal without and S type as an heat killed, and both he introduced into mice, and he found that the mice die due to pneumonia. He was very confused by looking at that. Given he only used R type, the mice is alive. When he used S type, mice died. But when he used heat killed S type, the mice is alive. But when he used both property, which in his first experiment shows living condition, when he used those two living conditions of bacteria, the mice will die due to pneumonia. So he was very confused, and he concluded that might be some properties are changed inside the R type, and he will transform into S type and may cause pneumonia. Let us discuss its conclusion. He says that the R-stain bacteria had somehow been transformed by the heat-killed stain bacteria. Some transformation principle. Transfer from heat-killed S-stain bacteria had enabled the R-stain to synthesize smooth polysaccharide coat and become virulent. So what he say? He says that if something from the S type migrate to the R type, and due to which R type produce a capsule around himself, and due to which it acquire the property of virulency, and it may cause pneumonia due to this capsule. And he conclude that further that transfer of matter which cause R type to produce capsule and convert it into S type virulence property is due to a material which is known as genetic material. Further, he conclude that it may be the protein which goes to the R type and produce a capsule over its surface. However, this capsule is made up of polysaccharides, so he conclude that maybe this create a polysaccharide over the that R type, and it acquire the property of virulency. However, this experiment is not concluded that the either property of genetic material or not. Later on, this experiment is further concluded by three scientists known as, one of them Oswald Avery, Colin MacLeod, and MacLean McCarthy. They performed this experiment many times from 1933 to 44 and finally conclude that DNA is the genetic material on the behalf of a biochemical testing, and that's why this experiment is known as biochemical testing of transformation principle. What they do, let us discuss one by one. They they centrifuge the material biochemicals. They centrifuge the biochemicals from the bacterial culture, and then they use three enzymes named as RNase, DNase, and proteinase, and protein degradation proteinase. These three enzymes are used by one and one principle like this. They you are seeing in Oswald and Avery experiment that they used heat gas type, and they separate by centrifugation the different chemicals of the bacteria, biochemicals present in bacteria or biomolecules present in bacteria. First of all, they add proteinase, so that there will be no protein inside this bacterial tube, the tube, scientific tube which contain bacteria. They add is this to the S cell appears, add to the R cell. Okay. When they add the protein without protein to the mice, this the mice die with pneumonia. Mice die with pneumonia. When they injected both to the mice cell die with pneumonia. Later on, they use RNAs and to the R and S type of cell, and the mice also die with pneumonia. Same type of result they found. Again, they used DNase enzyme due to which DNA is damaged, totally lost from this suspension, and they injected it to the mice cell, and they found that the mice is alive and no pneumonia is caused. So they found that might be it is DNA who caused transformation. Means first they used proteinase which degrade the protein, and they inject the RNA as well as DNA into the mice and the R type, and they found that mice will die with pneumonia. That's why it is clear that the protein does not cause any change. Again, they used RNAs and proteins and DNA are in living form, and mice again got the pneumonia and die. But when they use DNAs, there is no presence of DNA, and they use R type with this, and mice die with pneumonia. No pneumonia is caused in this case, and due to which mice is alive. It shows that if the DNA is transferred, then mice must be get pneumonia. But when DNA is not transferred, so R type is not converted into S type, and due to which no pneumonia is caused and mice is alive. It clearly shows that that material which transfer from the S type to R type is DNA, and it transform it into S type. Further on, so it is clear from the biochemical test which is performed by Oswald, Colin MacLeod and McCarthy. Later on, another experiment is performed by Hershey and Chase. Hershey and Chase in 1952 to 3 to further confirm that the DNA is the genetic material which is known as the radioactive isotope experiment. So this experiment is known as the radioactive experiment or Hershey and Chase experiment. So what they do, they use the isotope of uh, radioactive element phosphorus and the sulfur. As we know that today's that the DNA contain phosphorus as a backbone and in phosphodiester bond. And similarly, we know that the proteins contains sulfur. Protein contains sulfur and DNA contain phosphorus. So they use the radioactive S35 for the protein and the radioactive P32 for the phosphorus. As we already know, if you study in previous classes, that a bacteria process which is known as transduction, which is performed by 1954, 3 and 4 by Lederberg, and they find that the bacteriophages a tadpole-like structure of T2 or T4 bacteriophages attack on E. coli. Like this, they attached on the bacterial cell, produce a pore, introduce their genetic material, and yeah, they and produce a prophase by integrating their genetic material with the host genetic material and order it to perform the work for himself and produce their head, tail, etc. And later on they cause lysis. Later on they cause lysis of the cell. A similar experiment is performed, but now first of all they what they do first they use a one petri plate and feed the bacteriophage on P32. Some bacteriophages are feed on P32 like this, and make infection to this. Then by the help of blending, they separate the host or protein part of the bacteriophage, and his DNA is introduced inside this. Uh, this is, for example, the DNA of this. So this is the DNA of bacteriophage, and this is the DNA of bacteria. And they radioactive detect in the cell, and no radioactive detect from the supernet. After blending, they centrifuge the bacteriophage this particle, and they found that the radioactivity only present in the DNA sample. Radioactivity only present in the DNA sample of bacteriophages, all the bacteriophages which infect the bacteria. Similar experiment is performed for the S. They feed the bacteriophage on a S35 containing nutrient media, and same type the bacteriophage attached with the host body produced a hole inside the host body and introduced its genetic material inside the host body, and then they were with the help of blending they separate this mm, coal or ghost from the bacteria and centrifuge the bacteria to separate the bacteriophage and DNA molecules, and they found that the radioactivity only detached from the supernet. Radioactivity only detects because their DNA is not radioactively labeled, so radioactivity only detect from the protein part and not from the genetic material. Centrifugation technique is used to separate the biomolecules on the basis of their sedimentation coefficient. So in this way, the are separated from DNA, and DNA is separated from the other cell particles. So in this way, they use this bacteriophage, separate the bacteriophage from this protein code from already, and the DNA is mixed up. Bacteriophage produce their body. From that, they extract the DNA. Actually, they extract the DNA and found that DNA is radioactively labeled, and no protein is detached from this. No radioactivity detect in the protein part. That is clear. So it's mean that protein are synthesized inside this cell from the non-radioactive material, and here the protein is synthesized from the radioactive material. Okay, so this is all clear that the DNA is transfer from one cell to another cell as a genetic material. So these three experiment, one is Griffith's experiment, second is from the MacLeod Oswald Avery experiment, and the third one is from the Hershey and Chase have confirmed that the DNA act as a genetic material. Thanks a lot, and and kindly follow the channel, like it.